All right, so in this lecture, we're going to cover leukocyte circulation and migration into tissues. What I really like about this chapter is that it really starts explaining the complexity and genius, I think, of the immune system. And we see how it's not just a collection of individual cells that each have their own functions and act appropriately, but it's the networking, interlocking of these cells and the specific movements from circulation into tissue, back into circulation, or to the peripheral lymphoid organs, or from the central to the peripheral lymphoid organs in a very organized and specific manner. So it's very different from what I would describe the circulatory system, where blood flows in a predictable and continuous manner according to pressure gradients. These cells all know where they're supposed to be at a given time. So there's a few key terms that we're going to go over that we're going to be using a lot in this chapter. Okay, the first is just a description of the highly regulated movement of lymphocytes into tissue, which we're going to call migration or recruitment. And we say recruitment because typically, unless these cells reside or are born in those particular tissues, they're not going to be there for any other reason unless they're recruited to perform some sort of function. Then the opposite of migration or recruitment is recirculation, so the return of those cells back into the blood. And it's this kind of movement, the going back and forth, and not just being specific to a particular location, that is a unique property of the immune system. Now, migration and recruitment is just a general term. When these cells are going to a specific destination, which we'll learn as part of the inflammatory process or inflammation, we call that homing. So homing is just a more specific term of migration when you're referring to a very specific organ or part of the body. Now, the hallmark of inflammation is the 1-2 punch of the immune system, which is the innate immunity and adaptive immunity. So it's always first up is the innate immune response, which triggers, helps refine, and exasperate the adaptive immune response. So they always work together, and it's never just one by themselves. Now, these complex series of movements help to serve three functions. Okay, the first is the delivery of myeloid lineage cells from circulation into sites of injury or infection. And here's where I want you to think neutrophils and monocytes, which I know I use the term again, 1-2 punch of the innate immune system. Okay, and it's always the first responders, neutrophils, followed by the secondary responses, monocytes, within at the site of injury or inflammation, turn into macrophage, differentiate into macrophage. The second and third functions are functions of the adaptive immune response. Okay, so it starts off with the delivery of lymphocytes from their sites of maturation, like the bone marrow and the spleen for B cells, the thymus for T cells, to the sites of peripheral lymphoid organs. So we call those central, primary, generative lymphoid organs is where they mature, to peripheral, where they actually get antigen presentation. They proliferate whenever they find an antigen that matches to them, and then they further differentiate into effector and memory cells. Okay, then the third function is to deliver those effector lymphocytes from the site of peripheral lymphoid organs to the sites of injury or infection, where they can actually serve out their purpose. So first I'll give a general overview of the migration process, and then we'll get into the specific mechanisms of how all this works. Okay, so naive lymphocytes, so these are ones that have not encountered an antigen that's specific to them, are always in this continuous migration into and recirculation out of secondary lymphoid organs. Okay, so they constantly go in and out of these secondary lymphoid organs, just basically looking for something to do, okay, looking for that antigen. And these secondary lymphoid organs only have a certain capacity to hold a specific amount of cells. So these lymphocytes always have to go in, and then if they don't find an antigen, they exit, they circulate around, they migrate back into another lymphoid organ, see if there's an antigen. If it's not, they migrate out. Once they find that antigen and get activated lymphocytes then home into the tissues where they're recruited to. This is all the same for myeloid cells. They only migrate into tissues once they have chemical signals that initiate their migration into those tissues. Furthermore, memory lymphocytes will also migrate into lymphoid organs, mucosal tissues, skin, and other areas where they might reside on a more semi-permanent basis. Now, this whole migratory pattern into tissues requires adhesion of leukocytes to the endothelial surfaces, right? And it's a process that involves adhesion molecules and chemokine receptors and chemokines. So chemokines is a word that we're going to use a lot, and it's just a specific type of cytokine, which are sort of messenger proteins that these cells rely on to get their instructions. Now, the way endothelial cells recruit immune cells into tissues is that sentinel cells usually send out some sort of signal via cytokines that upregulate the adhesive molecules on these endothelial cells. So they become naturally more attractive to effector leukocytes, which then allow for migration into the tissue. Now, adhesive molecules can be divided into two categories, selectins and integrins. We're first going to talk about selectins, and these are carbohydrate binding adhesive molecules. In fact, they're a type of C-lectins, and the C stands for carbohydrate. So that means that they bind all carbohydrates and sugar polymers. In the adhesion process, they're the initial step, and they have a lower affinity than integrins. So in fact, they're designed to be continuously formed, broken, and reformed. Now, selectins can be divided in three categories. The first category we're going to talk about are L-selectins. L stands for leukocytes in this case. So they're expressed on neutrophils, monocytes, and naive B and T cells. And they're expressed in response to certain inflammatory cytokines, tumor necrosis factor, TNF, and IL-1. And we'll learn all about the functions of IL-1, TNF, and these other cytokines in the next chapter. But it's important to note how these selectins are expressed in various circumstances. Now, these selectins on the cells bind to various types of ligands that are found on endothelial cells. Okay, so it's important to note that whenever I say a selectin, that's it's not a receptor, but it's a surface molecule, and then the ligands are what get bound. Okay, so leukocytes express the L-selectins, and then endothelial cells have the ligands for these selectins on the leukocytes. And once we get to endothelial cells, we'll find it's the inverse of that endothelial cells have these other types of selectins, and the leukocytes will have the ligands on their surface. One notable example is that the HEVs, the high endothelial venules found in the lymph nodes, express high amounts of these ligands for the L-selectins. And it's really important so that T and B cells can migrate into them. The next two classes of selectins are both found on endothelial cells, and they are P-selectins and E-selectins. P-selectins get their name because they're originally found on platelets, hence P, but they're also found on endothelial cells. And then E, of course, gets their name because they're found on endothelial cells. So that L, P, and E is all ways of helping you identify where the selectins are expressed. And then you just want to think that the ligands for them are expressed on the opposite types of cells. Okay, so P-selectin is expressed in response to histamine, which we'll learn comes from mast cells, and then thrombin, a really important compound in the blood coagulation factor. Okay, P-selectin binds to ligands on leukocytes, such as neutrophils, monocytes, and activated T cells, just as ligands for E-selectin are found on neutrophils, monocytes, and activated T cells. Okay, now E-selectin, in this case, is expressed in response to cytokines such as IL-1 and TNF-alpha, which are expressed by sentinel cells like dendritic cells and macrophages, and then LPS, which is expressed by microbes. It's a very notable polysaccharide expressed and given off by microbes in foreign bodies. The second class of adhesive molecules I want to talk about are integrins. Now, in comparison to selectins, integrins have a higher affinity, so they form stronger bonds, and also more lasting bonds than selectins. Now, leukocytes express more than 30 different types of integrins, so I'm only going to touch on a few of them that pop up in the rest of the book. So the first one is leukocyte function associated antigen 1, which binds to ICAM1. So the ligand is intracellular adhesion molecule. So while this integrin is really important just for the general migration and recirculation of leukocytes, it also has a particular importance for the interactions of T cells and antigen-presenting cells. So this adhesive molecule facilitates these type of cells' interactions so that T cells can go from naive, interact with their antigen, and become activated. Another notable one is the very late antigen 4 and its adhesive molecule, vascular cell adhesion molecule 1, VCAM, which is just expressed on cytokine-activated endothelial cells, so it helps these cells migrate into sites of inflamed tissues. And then a third one I want to bring up is MAC1, which is expressed on neutrophils, monocytes, and dendritic cells, in general, most phagocytes. And while it does bind to ICAM1 ligand, its importance in phagocytes is that it also binds to opsonins, usually from the complement system. So it acts as a complement receptor to enhance and facilitate phagocytosis. So we learn about complement, I think, in chapter 9 or 10, a little bit later in the book. So this pops up, okay, the MAC1 and ICAM1 interactions. So integrins rapidly increase their affinity for ligands in response to chemokine and chemokine receptor binding in all leukocytes, okay, and also by antigen receptor binding in T cells. And they do this in two ways. One is that they change their conformity in order to increase affinity. So as we can see here in the diagram, they're usually found in a low affinity bent state where it's really difficult for them to interact with their ligands. But whenever activated, they extend and increase their affinity. The second way is that they also change their distribution to congregate in clusters on the cell surface as opposed to being evenly spread out. This helps increase the concentration of them and thus increase their total affinity for a group of ligands. Next up, we're going to talk about chemokines, right? So I've already mentioned these a number of times, both in the previous lectures and this lecture, so now I can formally describe them. So chemokines are a large family of cytokines that stimulate leukocyte movement and regulate migration of leukocytes from the blood to tissues. These polypeptides are about 8 to 10 kilodaltons in size, and humans have 47 different classes of chemokines. And these classes can be divided into 4 families, they're called, named after the cysteine bridges. So we have CC, CXC, the C family, and then finally the CX3C family. Now I can tell you that about 90% of the chemokines we're going to discuss fall in the first 2 categories, CC and CXC. So these are the 2 dominant families. And furthermore, a pro tip is that whenever there's an R or an L follows after them, followed by a number, the R stands for receptor and the L stands for ligand. So for instance, we might talk about the CCR7 chemokine. So that's a receptor, and there's 6 other receptors before that and a few other afterwards. Or we could say, as an example, the CX3CL21, which is a ligand because of that L. So whenever you see that nomenclature, now you can start to get an understanding of what type of chemokine it is and if it's a receptor or a ligand. Now, chemokine receptors belong to a G protein coupled receptor family, and they're what's behind the mechanisms that act for adhesion in integrins and their ligands. Okay, so chemokine receptors, in response to their messenger chemokines, signal cells how to activate or increase the affinity of their integrin receptors so that they can adhere and migrate into the tissues of where they're being recruited. And it's the different combinations of the chemokine receptors and their diversity that are expressed on the various types of immune cells and leukocytes, and their combination that mediates their distinctive patterns of migration and recirculation. So just a few quick tips on chemokines. All leukocytes express chemokine receptors. T cells express the greatest diversity, in part because they have to migrate to so many different areas of the body. And then there's also atypical chemokine receptors, ACKRs, which don't themselves engage in signaling pathways, but they inhibit or terminate chemokine responses in cells. So they sort of act as an off function for certain chemokine receptors. There are 3 primary functions of chemokines. The first plays a role in inflammatory reactions. Here, chemokines serve to recruit circulating leukocytes from blood vessels into extravascular sites. And this happens kind of in 2 separate steps. The first is just the natural increased adhesion by the increase of affinity of integrins on the endothelium. So this is on the walls of the blood vessels. So that helps leukocytes get a stronger and firmer adhesion to the walls of the endothelium. And then there's the migration of leukocytes from the endothelium into the extravascular tissues. That follows a concentration gradient of both the chemokines indirectly, which directly increase the affinity of the adhesion molecules as the leukocytes get closer and closer to the primary site of inflammation. Second, chemokines are both vital for the development of lymphoid organs, and then they also, within the lymphoid organs, regulate traffic of lymphocytes and other leukocytes through the different regions of those lymphoid organs. Now, in contrast to inflammation, where the affinity of integrins changes in response to inflammatory cytokines, chemokines present in the secondary lymphoid organs are homeostatic, which means they're expressed constitutively, meaning that they never change. Now, third primary function is that chemokines are required for the migration of dendritic cells and other antigen-presenting cells from the sites of infection into the draining lymph nodes. So, for instance, one that we're going to hear throughout this course is CCR7. So, dendritic cells express CCR7 receptor, as do naive T cells. And this helps them both congregate in the same areas of the lymph nodes. I always remember this for two reasons. I'm a big Creedence Clearwater Revival fan, so CCR. And then I think of Cristiano Ronaldo, the famous soccer player who wears the number 7 jersey. You always see CR7 as abbreviation for him. So it's kind of a mixture of those two. So the first step in cell migration into tissue involves sentinel cells like macrophages or dendritic cells at the site of infection, recognizing foreign microbes and releasing inflammatory cytokines like TNF or IL-1. These inflammatory cytokines cause a dilation of the blood vessels and a decrease in general flow of blood, which results in leukocytes being larger than red blood cells, moving away from the central axis of the blood vessels and closer to the lining and the endothelial cell layer, a process called margination. These inflammatory cytokines also cause an increase in the expression of EMP selectins on the endothelial cell layer. So remember, endothelial cells express these, and then the leukocytes have the ligands for them. And note in this picture, too, in the legend, that these cells have some inactive integrins, in part because they have yet to really experience the cytokines, the inflammatory cytokines themselves. Yet the endothelial cells, being closer to the site of inflammation, have experienced those cytokines. So that's why they express those. And this low affinity binding of the selectins to the leukocytes is constantly formed, broken, and reformed along the flow of blood. So it creates a sort of what we call rolling adhesion. So these cells are now closer to the endothelial cell layer. They loosely bind to the selectins. They break, they loosely bind again, and it constantly has the cells rolling along the blood vessel lining and also along a concentration gradient of selectins on the cell wall. As the cells approach the site of inflammation, chemokines start expressing or causing an increase in expression of integrins, which have a higher affinity than selectins. So here in the picture, we see ICAM1. We also saw VCAM1 and their respective ligands on the leukocytes. So these become activated. We have a firmer adhesion state, and the cells start even flattening out as more and more of their integrins attach to available ligands on the endothelial cell layer. Now, once this binding hits a critical point, the actual attachment of ICAM and VCAM1 on the cell surface starts signaling intracellular processes that instruct the cell to undergo paracellular transmigration. So that's when the leukocyte actually kind of disrupts the endothelial cell layer junctions and goes around those cells, those lining of the blood vessels, into the tissue. So that's an important difference actually between integrins and selectins, in that selectins are just surface adhesive molecules, whereas integrins, not only being firmer and having a higher affinity to their ligands, also have certain roles in intracellular processes. So they themselves act as receptors, just like what we learned with the MAC1 and the phagocytic receptor and ligand. Also point out here that this is just a general description of migration. So what tissues, what state of inflammation, and what cells enter at what points is all unique to every cell, okay, and what chemokines are expressed. Now, in the history of immunology, the importance of these selectin steps, integrins, and chemokines in all leukocyte migration patterns were first determined from gene knockout mice. So a big one was the CC18 knockout mice, so these mice had the CC18 gene knocked out of them, meaning removed, which resulted in complete deficiencies to develop many selectins and integrins on the cell surface. And then later on, we found out that there are humans that had rare inherited diseases that also had this gene missing. This disease is called leukocyte adhesion deficiencies. And as we can imagine, if the cells are unable to migrate into tissues, a whole host of symptoms can occur in these, such as recurrent bacterial and fungal infections because they don't have the cells necessary. They have the cells necessary, just those cells are ineffective in getting to the sites of inflammation to work effectively. A lack of neutrophil accumulation at injection sites, and then defects in adherence-dependent lymphocyte functions. So now that we've discussed general migratory patterns of all leukocytes, we can start getting into the individual migratory patterns of these different cells. Okay, so neutrophils and monocytes, as we learned, are phagocytes and are primarily found in circulation. But despite that, most of their phagocytic effector actions take place within tissues virtually everywhere in their body. Okay, so they're pretty much in an off state as they're circulating, and it's not until chemokines direct them to migrate and they're recruited into sites of tissue where they actually perform their effector functions. This is in part because chemokines and other inflammatory cytokines are what initiate the action of phagocytosis. So they're not usually found just in the blood, unless that is actually the primary site of infection. So both these cells follow the same general steps. It's just what chemokines and what ligands are expressed that leads to their different both timescales and mechanisms of actions. Okay, so neutrophils, as we learned, are the first responders. And this is because there's an early abundance of the CXCL8 chemokine. And as the abbreviation suggests, that's ligand that's released by tissue-resident macrophages and other sentinel cells at the infection sites that binds to the neutrophil-expressed CXCR8. Okay, so it's really easy to remember those two because they have the same abbreviation, and except one is L, one is R. Monocytes, because they become later, as you can guess, that chemokine gets expressed later. Okay, so it's the production of CCL2, which binds to only the monocyte-expressed receptor CCR2. So a nice, really symmetrical mirror between those two, CCL, CCR, and they both have 2, neutrophils have 8. So that occurs in a later step. So neutrophils and monocytes were really easy. That was a nice, simple slide. Now that we get into lymphocytes, it gets a lot more complex. But I promise you, it's not that hard. Okay. So first, we're going to concentrate on T cells. So when mature, naive T cells emerge from the thymus, they home towards the T cell zones in the secondary lymphoid organs. And as we discussed before, T cells are in a constant state of migration into secondary lymphoid organs, short residence time in the lymph nodes, and then recirculation, so the return back in the circulatory system, a process we call lymphocyte recirculation. And this is what helps T cells hone in and find the correct antigen if there is one there to present. So in fact, there's a high amount of traffic for T cells. Lymph nodes have enough flux of 25 billion cells per day. So every lymphocyte in the body goes at least through one lymph node per day. Okay, so you can imagine why it can take so much longer for an adaptive immune response to occur in comparison to a native immune response. But still, the fact that there's this high amount of traffic almost guarantees that T cell will see its antigen within a day if it's in a lymph node. And there's other few processes that help promote this. So for instance, during inflammation, there's an increase of blood flow through the lymph nodes, which brings with it an increase of T cells as well. And then there's also a limiting of the egress of T cells from the lymphatic system. Okay, so that limits and increases the amount of time of T cells within the lymph nodes, and that increased residence times also increases the chances that T cell is going to see its antigen. Now, the homing of naive T cells into these lymph nodes occurs through a few processes, and we learned that the entry point is through these high endothelial vesicles, HEVs. Okay, so it starts with the selectin binding, which is the weaker binding of the two adhesive molecules. So the L-selectins on T cells, remember L stands for lymphocyte, binds to the ligands expressed on HEVs. The subsequent step is a firmer adhesion through integrins. We learned that LFA, and again, think of that L for lymphocyte on the cells binds to ICAM1s, also VCAM1 to a limited extent on the HEVs. And the chemokines that help produce this and help promote this is CCL19, CCL21, those are the two ligands. Again, think of that L for ligand, produced by the fibroblast reticular cells and HEVs that bind to CCR7 on T cells. Now, CCR7 is a receptor, a chemokine receptor that's going to come up a lot. So I definitely want you to commit that to memory, its respective ligands. And it's also going to be really important because dendritic cells express the same exact one. And as we're going to learn in subsequent chapters, the interaction between dendritic cells as an antigen-presenting cell and T cells is vital for T cells in order to recognize their antigen and then perform their effector functions. This is a nice little picture, by the way, in the bottom right-hand corner of T cells actually migrating through the luminous surface of HEV. So all those white circulars are T cells traveling through the lymph nodes. Now, at this point, CCR7 and its respective ligands, CCL19 and 21, has done its job to get T cells and dendritic cells localized in the same lymph node. But there's still a pretty monumental challenge in that there are thousands of both naive T cells and dendritic cells trying to meet up with each other. And remember, T cells is that rare T cell that is unique to that one antigen that some of these dendritic cells are trying to express. So the challenge remains, how can we get these two cells to interact with each other? It's just like trying to find your friend at an extremely crowded bar. All right, so the first step is that fibroblast reticular cells, FRCs, form these 3D networks that traverse the entirety of the T cell zones, and they're covered with the antigen-carrying dendritic cells, right? And they also secrete CCL19 and 21, which helps promote the migration of T cells across these 3D networks. And the T cells are surprisingly really fast. In fact, they move at this amoeba-like speed of 12 microns per minute, which for a cell is insanely fast. Right? So the dendritic cells are stationary throughout these networks. And because of this, a single dendritic cell that's stationary might encounter around 85 T cells per minute. And a single T cell may remain in a lymph node for up to 24 hours. So there's a really high chance that these T cells are going to find a dendritic cell expressing that antigen that's specific to them if it's there. And if not, then they exit from the lymph node. Now, the exit, and actually indirectly the entrance of naive T cells into lymph nodes and out of lymph nodes, is mediated by a lipid chemoattractant S1P. Okay, S1P is a protein that's abundant in the blood in circulation and is lacked in lymph nodes. So cells within circulation, due to overexposure to S1P, start suppressing their S1P-R1 receptor. Okay, and S1P helps cells migrate towards a concentration gradient of S1P. So once they lack that receptor due to overexposure, they're kind of tired of it. That allows one to leave circulation and enter the lymph nodes due to the chemokines that we just learned about previously. Once in the lymph nodes, because there's a lack of S1P, cells start to re-express that receptor, the S1PR1, within a few hours. And it's this sort of lag time in between underexpressing and then re-expressing that helps the T cells remain within the lymph nodes. So after a while, if they haven't encountered their antigen, they're going to start expressing regular amounts of the S1PR1 receptor and then travel towards the concentration gradient of that lipid, which is again in the blood. So that's what promotes the exit from the lymph nodes. All right. Now, if cells were to find their antigen, activation actually suppresses the re-expression of that receptor. So even if they haven't seen it in a few hours and they are activated, they're not going to re-express that receptor. And that allows them to remain within the lymph nodes, undergo clonal expansion and differentiation. Now, the mechanism for the suppression is a protein, CD69, that gets activated on the lymphocyte cell surface once that cell is activated. And this protein binds with S1PR1, inactivating it. So those cells would still have in that receptor, but it would also be inactivated. Now, once activation is complete and the cells have expanded, there's millions of copies of them, then that protein will that process will reverse. That protein will not be expressed, and then those cells are then allowed to express activated and fully functioning forms of S1PR1, allowing those cells to exit. Now, in previous studies, scientists have shown the importance of S1P and the S1PR1 receptor in knockout mice that lack the gene to produce this protein, this lipid, by showing that naive T cells taken from these knockout mice, even when injected into healthy cells, enter the lymph nodes and are unable to exit. And this, in fact, led to the development of fingolimod, a drug by Novartis that acts as a S1PR1 suppressor. So people would take this, have really aggressive T lymphocytes that would normally enter the blood-brain barrier and attack cells of the central nervous system, and that's what multiple sclerosis is. So when taking this drug, it suppresses the expression of that receptor, and these overly aggressive T cells go in within the lymph nodes and then stay there for longer periods of time. In fact, there's a really nice video that I'm going to cut to now that helps explain S1P perhaps a little bit better with nice visuals, and then we'll get back to the presentation.
Multiple sclerosis, or MS, is one of the most common disorders of the central nervous system in adults, afflicting up to 2.5 million people worldwide. The pathological process involves circulating autoaggressive lymphocytes that cross the blood-brain barrier into the central nervous system, which leads to inflammation, tissue damage, and ultimately gliosis. These sites of tissue damage can be visualized as lesions on MRI scans. Sphingosine-1 phosphate, or S1P, is a naturally occurring bioactive sphingolipid that plays a key role in MS-relevant processes, including inflammation and repair. S1P1 receptors are widely expressed on lymphocytes, and these receptors regulate the normal egress of lymphocytes from the lymph nodes. During egress, T lymphocytes migrate across an S1P gradient that exists between lymph nodes and the efferent lymph. During a normal protective immune response, the activation of T cells in the lymph nodes causes an internalization of S1P1 receptors, and this prevents their immediate egress to allow optimal activation and differentiation. At the end of the differentiation process, The T cells upregulate S1P1 again, and they egress into the efferent lymph and blood to recirculate to peripheral sites. In patients with MS, circulating autoaggressive lymphocytes cross the blood-brain barrier into the central nervous system, where they cause inflammation and tissue damage. The resulting inflammation destroys the myelin sheets that surround the axons, and oligodendrocytes are lost in great numbers, leading to demyelination, along with axonal and neuronal loss and gliosis. The central nervous system can repair itself after injury. However, upon repeated inflammatory attacks, these endogenous repair mechanisms become less and less successful because of the inhibitory environment within the lesion sites. Thus, there is some recovery in the early stages of the disease. But following recurrent inflammatory attacks, the repair mechanisms become less effective, and attacks increasingly cause tissue damage and gliosis. Current first-line standard treatments primarily target the inflammatory component of the disease and are partially effective. Novel treatments are needed that can directly target the central nervous system and involve neurodegenerative and repair processes, in addition to reducing inflammation. Oral fingolimod is the first molecule in a new class of S1P receptor modulators that is being developed for the treatment of MS. After oral administration, fingolimod is absorbed into the bloodstream. Once in the bloodstream, the drug becomes biologically active, following endogenous phosphorylation by an enzyme called sphingosine kinase 2. Fingolimod phosphate readily enters lymphoid tissues, where it binds and downregulates S1P receptor type 1 on lymphocytes. This process abrogates migratory responses of lymphocytes to S1P and reduces the numbers of cells that egress from lymph nodes back into blood. In multiple sclerosis, fingolimod retains autoaggressive lymphocytes in the lymph nodes, away from blood and the central nervous system where they would otherwise produce inflammation and tissue damage. The retention of blood lymphocytes in the lymph nodes following the administration of oral fingolimod does not represent a significant burden to the lymph nodes, because lymphocytes are usually present in high concentrations in the lymphatic system, with a comparatively tiny amount in the bloodstream. Oral fingolimod does not affect peripheral effector memory T lymphocytes, as they localize in non-lymphoid tissues without regular recirculation through the lymph nodes. The sparing of effector memory T lymphocytes may be important for immunosurveillance and memory immune responses in the peripheral tissues in patients treated with oral fingolimod.
Now, once activated, T cells have clonally expanded, differentiated, and have become effector T cells or perhaps memory T cells. And they still have to exit the secondary lymphoid organs in order to act as sites locally of infection. Okay, so the challenge remains how this happens. And much of the same way that this happens mirrors how they stayed, or is the exact opposite of those procedures, or what caused them to enter the lymph nodes in the first place. So the first step is that S1PR1 expression is restored after proliferation, differentiation. So now cells want to go back into circulation where S1P is more abundant. They lose their expression of CCR7. So they're no longer going to fall into that recirculatory pattern because they no longer express CCR7. And they start expressing receptors for inflammatory cytokines, just like neutrophils and monocytes would. They also stop expressing L-selectin, and they start expressing ligands for E and P selectins. Again, just like monocytes and neutrophils. So they're no longer going to start that pattern of migrating into lymph nodes because of the lack of L-selectins. In this case, the E and P selectins is going to make them migrate and home into the sites of inflammation. Now, memory T cells act a little bit differently than effector T cells. So they might go in one of two places. One is that they can go and migrate to sites of tissue where they might be more effective, like the endothelial layer of the gut or the skin. And they will reside there and respond rapidly by producing cytokines whenever they re-encounter their specific antigen. Or they could become central memory cells and remain within the secondary lymphoid organs, where they're going to proliferate and activate much faster than their naive counterparts. Now, as I mentioned before, T cells express the most diverse set of chemokine receptors because they have to migrate to many different parts of the body. B cells, less so much. So B cells is going to be sort of a simplified version of T cells. They follow many of the same patterns, but they don't have to go to as many nearly tissues because their effector functions are to produce antibodies, which act at many different sites of the body, as opposed to the cells themselves, as in the case of T cells. So again, it follows the same basic mechanisms as T cells as for entry and exit of the lymphatics. As we learn, B cells leave the bone marrow and they mature in the spleen. They do this through the CXCL13 ligand, and then the receptor here is CXCR5. So you just have to remember that the L13 ligand responds to the R5 receptor for some reason. It's not as symmetrical as some of the others. So this helps complete maturity. The fully mature follicular B cells re-enter circulation through the same process as T cells. It's an S1P-driven mechanism. Naive B cells enter the lymphatic system through rolling adhesion and the HAVs, chemokine activation from soft to firm adhesion, process just like T cells. So we're going to see a lot of the same chemokines here, CCL19, CCL21. Again, I want you thinking CCR7, if it helps you remember. Again, my way of memorizing that is Cristiano Ronaldo wears the number 7 jersey, and Creedence Clearwater Revival, CCR7, on naive B cells. Furthermore, the CXCL13, produced by these follicular dendritic cells, helps bind to the CXCR5 receptor in naive B cells, which guides them into the follicles. That's where B cells meet their antigen and differentiate into plasmablasts, effector B cells, or memory B cells. Now from there, different subsets of B cells go into different types of the body, and different ones produce different antibodies, which we'll learn about, I think, in one of the later chapters. But these long-lived, fully differentiated plasma cells, called plasmablasts, are what produce these antibodies. So ones that reside in the bone marrow tend to produce IgG antibodies, and then ones that reside in the mucosal-associated lymphoid tissues will produce IgA. So we'll learn all about the different functions of the antibodies later, but that's a nice simplified version of B cell migration. Again, it follows a lot of the same mechanisms as T cells. There's a few different chemokines in here, CXCR5, for instance, that brings B cells in the follicles rather than the T cell zones. But other than that, it's residing in the secondary lymphoid organs or the other lymphoid tissues, where they then produce antibodies that act at various sites, as opposed to T cells, which have to go into all the different other tissues or sites of inflammation directly. All right.